In this video I will discuss the development of the thymocytes, T cells. T cells are produced in bone marrow. Suppose this region represents bone marrow, and then from the bone marrow the T cells will enter thymus, and this represents the thymus. The development of T cells occur in the thymus. Thymus is divided into different anatomical regions, and as the T cells develop or thymocytes develop, they move from one region to the next region. Progenitor T cells that are produced in bone marrow will enter the thymus through the high endothelial venules and migrate to the subcapsular region of the thymus. So they are produced here, and then they will enter the thymus and they will reside or migrate to this region. Those progenitor cells or thymocytes will start or initiate their process of maturation by first rearranging their gene for the T cell receptor. So the T cell receptor genes are rearranged, and that initiates the process of maturation of the T cell. At this stage of the development, the T cells are double negative, indicating or means that they do not express CD4 and they do not express CD8 receptor. And as this gene rearrangement occurs, they will express both CD8 and CD4 receptor, and then they would be called double positive. So this double negative cells move toward the cortex, toward the medulla as they develop, and they will express both CD4. Now that they express both, suppose this red one over here indicates CD4 and this blue one here indicates CD8. Now the cell expresses both CD4 and CD8 receptor, and we refer to this thymocyte as double positive because it has both CD4 and CD8 receptors. And that's still occurring where? In the cortex. And in the cortex, one more thing occurs as positive selection. Positive selection will determine whether the T cell receptor, which is CD8 or CD4, could bind either MHC class 1 or MHC class 2 proteins that are found on the surface of the cortical epithelial cells in the cortex. If they could even weakly bind to any of those MHC class 1 or MHC class 2, they're okay, they're selected as a positive select cells. If they cannot bind to MHC class 1 or MHC class 2, then they're not selected. They cannot go through this process of positive selection. So positive selection will determine, in a simplified way, whether this thymocyte could either bind to MHC class 1 or MHC class 2. So if you, at this point, if the cell recognizes MHC class 1 protein, then it will receive survival signal and maturation signal, and this cell will express only CD8 and stop expression of CD4. And on the other hand, if the cell recognizes MHC class 2 protein, then it will receive survival signal and maturation signal and maintain the CD4 receptor and stop expression of CD8 receptor. So either the cell will become a CD4 or a CD8, and now those cells are in the medulla, and those cells are referred to as single negative. So we have either single positive cells or single I'm sorry, here are the cells double negative, they don't have any of the receptors. Here the cells are double positive, they went through the positive selection, and after that they will express either CD4 or CD8 depending on what kind of signals they get and what MHC class proteins they recognize. If they recognize MHC class 1, then they will express CD8. If they recognize MHC class 2, then they will express CD4 and stop expressing the other receptor. At this stage, the cells will go through the process of negative selection. And what is negative selection? The negative selection is if any of those cells show an immune response to self-protein or bind strongly to self-protein, then those cells will be eliminated, because it means that those cells will eventually attack self-protein, your own cells. You don't want that to happen. You want to eliminate those cells. So to recap, cells are produced in bone marrow. They migrate to the cortex of the thymus. They're double negative cells. They do not express any of the receptors. They begin their expression of the messenger RNA, and the T cell receptors are expressed. Those cells are still in the cortex. They're double positive. They express both receptors. Then they're gonna bind to the endothelial cells. They will either bind to MHC class 1 or MHC class 2. If they bind to MHC class 1, they will express one type of receptors, maintain the expression of one type of receptors. If they bind to the MHC class 2, they maintain the other type. And in here, they're going to go through the process of negative selection. And those cells over here are referred to as single negative. So you have double negative, double positive stage, and single negative stage, where they express either CD8 or CD4. In the next video, I'm going to explain how they differentiate into T cytotoxic cells. The CD4 cells differentiate into different types of T cells, like T helper cell, T cytotoxic cells.